Hello, everyone. It's a pleasure to be speaking to you all about some of the work we've been carrying out in the lab. We're a lab that's interested in the process of membrane fission. And I'd quickly like to get into telling you a little bit about what this process of fission means. This is a phenomenon that manifests quite elaborately and dramatically when you talk about the process of vesicular transport. And this is a process where newly synthesized proteins in the endoplasmic reticulum of the cell package these proteins in the form of vesicles and transfer it to the Golgi apparatus. From the Golgi, you release another set of vesicles. And these vesicles eventually end up going to the plasma membrane and fusing with the plasma membrane. And that seems to be the preferred route by which the cell ends up displaying the right sets of proteins onto the plasma membrane. Now, for this process to take place, the cell obviously has to produce a large number of vesicles and has to display a sustained activity in production of these vesicles. And the final act of releasing a vesicle from a donor membrane compartment is termed as the process of membrane fission. And in order to understand this process, we've been working towards understanding two aspects that eventually lead to fission and release of a vesicle from a given membrane compartment. The first pertains to how you end up defining a bud-like structure on a planar bilayer. And the second pertains to how you clip the neck of this bud, thereby releasing a vesicle into the cytoplasm. And the kind of questions that we've been asking pertain largely to understand the basis for specific sets of proteins that appear to catalyze both the budding and the fission step leading to vesicle release. And in order to address these kind of questions, we've been devising assay systems to critically analyze the process of membrane fission. And in order to do that, we've been spending quite a bit of time devising what are called model membrane templates that sort of mimic different stages of this process of membrane budding leading to fission. And if you look at a traditional vesicular transport reaction, as is shown here in a series of electron micrographs on the left of the slide, you'll see that the process starts by the planar bilayer undergoing some form of invagination or budding. And this bud in turn ends up defining a narrow neck. The neck is what connects the bud to the parent planar membrane. And through a process of fission, this bud then gets clipped and the vesicle is released into the cytoplasm. And if you look more carefully, then the bud is connected to the plasma membrane by a neck, as I said before. And the neck is nothing but a hollow cylinder of membrane. And if we were to be able to mimic this topology of the membrane, we thought we'd at least be able to critically understand the process of what sets of proteins act on this tubular cylindrical structure in order to lead to membrane fission. So in order to make membrane tubules, we resorted to an assay where we start with a passivated glass cover slip and spread some lipids on one end of that cover slip. These lipids are typically suspended in an organic solvent. We dry the organic solvent and place the cover slip inside of a flow cell and fill the flow cell with buffer. And the moment the dry lipid sees buffer out of a spontaneous process driven primarily by the hydrophobic effect, the dry lipids end up forming these large vesicles. And then you use the same flow cell setup to flow buffer at high flow rates. And the shear force exerted by the flow of buffer onto these vesicles extrudes these vesicles into very long, narrow membrane nanotubes. And the region where the lipid was spotted transforms into a supported bilayer. And downstream of this, you essentially end up forming an array of membrane nanotubes. And from a process that relies on very simple quantitative microscopy, since you already have a supported bilayer, you can equate the area you can create the fluorescence that it emanates from that's supported by LEP, provided you dope the lipid mix with a little bit of fluorescent lipid into the size or the dimensions of these tubes. If anyone is more interested, then I encourage you to look up this reference where we describe this whole process in great detail. With these tubes that are sort of are laid out on a passivated glass cover slip, these then, as a system, this automatically becomes amenable to microscopy. And one of the things that we've been trying to understand is what sets of proteins could get recruited onto these tubes. And in the process, also be able to understand what these proteins are capable of doing to this tube. So as a proof of concept, we decided to go back to this vesicular transport reaction. And we know from work from a large number of groups that at least one form of vesicular transport relies on recruiting a protein called dynamin to the necks of these budded vesicles. And dynamin appears to then use the energy release from GTP hydrolysis to catalyze fission. So we asked if the assay system indeed reports on fission, then could we recreate the process of at least the dynamin catalyzed fission using these assay systems? And to describe to you in greater detail the potential of this assay system, this is a typical field of the microscope after these templates are prepared. You'll see these parallel lines are, each of these are a long membrane tube. You'll also notice that the tubes are, they display differences in their brightness, and that's largely because of the size of these tubes, because each of these are diffraction limited. A thicker tube will appear brighter under the fluorescence microscope than a thinner tube. Now, the assay system is fairly stable, and one of the reasons why that happens is with time, these tubes, which are initially in solution, slowly settle down and get pinned at discrete sites along the length of the tube onto the glass cover slip. Because all of this is housed inside a flow cell, it makes it very convenient for us to flow in the right sets of proteins and assay for what they are capable of doing to these membrane tubes. So this particular experiment displays flowing in dynamin onto these tubes in the presence of GTP. And dynamin here is unlabeled. And as soon as dynamin and GTP reaches this particular field under the microscope, you start seeing each of these tethers getting cut into tiny bits. You can then obviously record this as a movie, go back and play this back in time, and start asking very interesting questions as to what caused the process to take place. Can we now start probing functional details of this reaction more carefully? Can we start understanding conformational changes that could have been elicited in dynamin that sort of led to this kind of a reaction manifesting on these tubes? So in some ways, this validates our assay for membrane fission. And because of the simplicity of this assay, one of the questions that we have been interested in is going beyond the dynamin paradigm and asking questions as to what all sets of proteins are present in a typical eukaryotic cytosol that could catalyze this reaction of membrane fission. And for that, we use this assay system in a screen. The reason to think of these along these lines is because of how straightforward the readout is. You start with a template which has these tubes, and if you were to add a lysate, a cellular lysate, for instance, would the tubes get severed? And you basically score for the percentage of tubes that get severed, and that's a fairly straightforward readout. So we've been, over the years, we've been looking at sources of proteins from a wide variety of organisms. We largely have restricted our focus to understanding the sets of proteins that are present in something as simple as a fungi to something as complicated as mammalian tissues. And the basis for the screen is fairly straightforward. We start with either cells or tissues, and we mash them up, extract the lysate out of them, and we then process the lysate in a certain manner, which I'll talk about in more detail in subsequent slides, and ask if the lysate has any set of proteins that is capable of catalyzing fission. So just to give you a glimpse of one sort of workflow that we've been relying on for a couple of years, it is to start with a native lysate, homogenize them, and give them a high-speed spin. And this essentially pellets down all the membrane fractions in the lysate. And we pass the lysate onto a desalting column. This essentially gets rid of all small molecule metabolites and nucleotides, and then resolve these proteins in the lysate using an anion exchange chromatography, and collect separate fractions from this chromatographic separation. And we pass each of these lysates onto these tethers. And because the lysate has been removed of small molecules, you now have the advantage of adding in defined conditions or adding in defined sets of small molecules, and ask, is there a reaction, a fission reaction that's either sensitive to GTP or ATP, or works just as well in the absence of any of these nucleotides? So what I'm showing you here on this slide is work from a number of students in the lab. We basically, what's plotted for each of these plots, what's plotted on the x-axis is that fraction that was isolated through an anion exchange. And what's plotted on the y-axis is the percentage of tubes that get severed in these assay systems. And for three separate conditions: one is in the presence of GTP, the other is in the presence of ATP, and the third is in the absence of any nucleotide. Now, very simple biochemical logic indicates that you have at least three separate sets of activities. One that is GTP dependent, the other that's ATP dependent, and the other that doesn't require any specific nucleotide, which is kind of interesting because this right away opens up the field to asking questions beyond the dynamic paradigm. Like I said, the GTP dependent fission is dynamic dependent, and over the years we've been trying to understand this process in greater detail. The ATP dependent process, we published this recently, is dependent on a molecule that's sort of like a cousin of dynamin. It's contributed by a molecule called the EH domain containing ATPase, and we're trying to understand the cellular basis or the reason for evolving a separate ATP dependent fission catalyst or a fission protein, and how it contributes, how this function contributes to cellular physiology. And the third activity is a completely nucleotide independent fission process, which is kind of interesting because one then wonders how this kind of a reaction gets regulated inside of cells. It's still early days for us in order to be able to understand the nucleotide independent fission reaction. But hopefully I should be able to tell you more about this in a few months to come. So in summary, what our results establishes is a robust and facile workflow to understand extreme forms of membrane remodeling, which is primarily fission. And it sets up the, as you would have appreciated, hopefully, it sets up the basis for screening for proteins for this kind of an activity. As far as biological systems are concerned, this is kind of an interesting screen because since it's a screen that is based on following through with activity, it circumvents the problems of redundancy or lethality that are typical to a lot of cellular screens. And with these new sets of activities that we've discovered, one hopes to then go back and ask what this activity means for the cell. And the way to do this would be to knock out the separate sets of proteins and look for consequences to cellular function. This obviously opens up new areas in cell biology and also helps us understand the basis of life, because membrane fission appears to be fairly critical for sustaining life. With that, I'll come to an end, and I'd like to acknowledge all the people over the years who've contributed. Not all of them are shown in this photograph, but it's basically been kind of an interesting process, starting all the way from devising the membrane template and then going down this path of undertaking a screen-like approach to look at this process. I'd also like to acknowledge the funding agencies listed here on the slide. And lastly, I'd like to thank all of you for your attention, and I'll be happy to address any questions you may have about this work. Thank you.